So let us begin off with our today's session. Yes, so we will be dealing in detail about the fertilization today. Right, so this is going to be a very important subtopic for your preparation, because you know that many a times in your examination this very particular topic has been coming up, and it is a really rewarding as well as a scoreable topic for you, right? So don't miss out your preparation. Just commit yourself with this topic such that you can seriously learn a lot on this and you can apply to your examinations, right? So as far as welcome to our channel. So this channel is Unacademy live, and this exclusively meant for your CSIR net, right? So I am one of the educator and Dr. Saranya. I am teaching live for you in this Unacademy. Right, so I have already completed my PhD in biomaterials, and I do have 13 papers of very good impact with me, and I have a 10 years of teaching experience combined with the research, and obviously have cracked net with the AR of 52. Yes, everybody, with this introduction, now we will go into our topic for discussion today, that is on the fertilization as well as this polyspermy prevention. If you take this topic as such, it is a very rewarding topic. Why? Because if you scan through the pre-IQs, you can find that almost every year, right? I'll tell you, every year there is a question, okay, from this fertilization, right? So it is definitely highly repeating and highly rewarding. That is why you must study this topic. Right, so what we will be seeing today in our topic, it is on your urchin mechanism of fertilization, and then we will see a little bit on what is resact, what is binding, and how your resact and binding is going to interact with each other, right? So all those things we will be seeing today, and then we will see that what is your acrosomal reaction, what is a cortical reaction, as well as what is your fast and the slow block. Like this is what we are going to see today. So let us begin our class from the basics of how the sperm and the egg, okay, so it is going to interact. Yes, so this is what your total process of the fertilization. Yes, it talks about how your sperm and the egg it is going to interact with, right? So if you take that, the chemo attraction, okay, so this chemo attraction of the sperm to the egg, okay, so how it is happening, how that sperm will know that the particular species of egg is there, only because of these soluble factors. So this is what we call them as your chemo attractant. Okay, so these chemo attractant are being produced by means of these eggs. Okay, so these eggs, they are going to produce these chemo attractant. By that, this fellow is going, this sperm is going to get attracted towards the egg, right? I hope that now you got the point of how your chemo attraction is taking place. Egg is going to produce some chemo attractant. With that chemo attractant, the sperm is getting attached or attracted towards it, right? So that is what it is called as your chemo attraction. Right, then what is going to take place? There will be the exocytosis. Okay, right. So there is going to be the exocytosis of your acrosomal vesicle, such that it is going to release its enzymes. Okay, so once the enzymes are getting released by means of your acrosomal vesicles, right? So this acrosomal vesicle is going to release all the enzymes. Yes, probably it contains many rich proteases. So once all these enzymes have been released, then what is going to happen? There will be the binding of the sperm, okay, to the extracellular envelope. Okay, so there is really going to be this binding of the sperm with your extracellular envelope. Right, so that is what it is going to take place. And then what is going to take place? See here, there will be the passing up of the sperm through the envelope, and finally it is going to lead to the fusion between the egg and the sperm. Right. So how the sperm is going to get attracted towards the egg? I told you that it is because of the process of chemotaxis. Why chemotaxis? Because no, the egg is going to secrete. What? It is going to secrete your chemoattractant. Right. So this Miller, he demonstrated. Okay, so he took off with this Cnidian orthophyxis. Okay, and he found that it is going to secrete a chemotactic factor. Okay, so that is going to regulate the timing of the release. What do you mean by timing? See, if you see an egg, does the egg, it is going to secrete this chemoattractant every time? Yes, do you think that the egg will secrete this chemoattractant every time? No. It has a specific timing of release. Okay. So probably, yeah everybody, welcome to the class. Probably whenever there is ovulation, that indicates that the egg is probably matured. Okay. So if this egg is probably matured, then only it is going to release all these chemoattractants or all these chemicals. Right. So that is what the timing is seriously very well managed of during this fertilization. It is not randomly released, but whenever there is a ovulation, only at that time it is getting released. Yes everyone? Right. So what happens? This scientist Miller, he fixed this developing oocytes at various stages. Okay, so let it be at the different stage he started to fix the cells, and then he added the sperm. Okay, so whenever these oocytes were not matured, okay, if the oocytes were not really matured, it was not able to attract the sperm. Right? Okay, but if the oocytes were matured enough, then it was leading to this attraction. Yes everybody? So you got it? This is how literally the eggs are getting attracted or attached towards the sperm. Right? Yes. So this is what you have to understand regarding the fertilization. Yes. So this is a very important stage. Right? So the timing at which it is approaching is seriously important. Right? So probably what will happen? So now the eggs are getting ready to be fertilized, especially during this ovulation condition, right? So after that, the oocytes, they control not only the type of the sperm. We already saw about the timing, isn't it? So one and only if your oocyte is mature enough, it is going to attract the sperm, or else this is not going to take place. Right. One more thing I was telling you, I am going to tell you is about the species. Okay, so if at all you have the oocyte of one species and the sperm of other species, so will it be able to interact with each other? Yes. So you have this oocyte of one species and the sperm of other species. So will it be able to interact with each other? No. Yes, it is not going to interact. Why so? Why it will not interact? This is because, yes, this is because of the fact that there is always the species specific interaction. Right? So that is what it is taking place. Always there is going to be a species specific interaction. This sperm can interact only with a definite oocyte. Okay, so it cannot interact with the any other sperm. Okay, so this particular sperm is meant to interact only with the particular oocyte. Yes everybody? So you got it? So that is how it is made to be still more specific. Right. So coming to this chemotactic molecule. So what is such a chemotactic molecule? So we have seen many times now, even today we have seen that chemoattractants, they are functioning to attract the sperm. So what is the best example of this chemotactant molecule? Yes, Resact. Resact, it is going to be the most important chemotactic molecule. Okay, so what is this Resact? Where it is produced? It is produced by the sea urchin egg, right? So the sea urchin egg, it is going to produce with the Resact. Yes, so this Resact, it is going to be a highly important molecule. So that is serving as a chemoattractant. Yes, everybody, I hope that till now it is all clear to you. We have just begin off with the fertilization and I have told you that these chemoattractants are going to be secreted. Okay, so by whom? Yes, these chemoattractants are secreted only by the egg. Okay, and if you take your sea urchin as an example, what is the chemoattractant it is secreting? It is going to secrete the resac, right? Fine. So will the resac be secreted every time? No. So whenever this oocyte is getting matured, only at that specific timing this is getting secreted. So can this resac interact with all this sperm? No. So if at all the sperm is from other species, it is not going to attract. Right? So these are all very basic points, but still it is important. So how does this resac knows that a particular sperm is coming? Okay, so it is going to have a receptor. So what is that receptor? This resac, it is going to bind with a receptor. That is what it is called as this bindin. So where should be the bindin present? Yes, everybody, resac, it is present in the oocyte, right? So where will be the bindin present? It should be present in the sperm. Okay, so this is the next important point. Right? So resac is present in the oocyte. So that is going to interact with the bindin. Where the bindin is present? The bindin is present in the sperm. Right. So this resac, it is acting very drastically. Okay. So because of this resac present, the sperm is getting activated. Right. So what it does? It increases this mitochondrial respiration. So the sperm is very quickly moving towards this oocyte. Okay. So now this oocyte, it is going to contain this chemoattractant molecule. So this chemoattractant molecule is giving a signal. And now this sperm is already, it is containing this bindin. Okay. So what it does, because of this chemoattractant, the sperm is binding very fast. So the sperm motility, it is increased so much. How it is increased? By means of the increase in the mitochondrial respiration. Why mitochondrial respiration? This mitochondrial respiration, it is going to activate and give this energy in form of ATP hydrolysis. Right? So who is doing that? See, this dynein ATPase is there. Okay. So this dynein arm you could have studied, this dynein arm, it is an integral part of the flagella. Okay. So now what happens? This mitochondria that is present inside the sperm, it is going to get activated. Okay. So because of that activation, it is going to trigger this dynein ATPase. So this dynein ATPase, it is going to release this ATP. So now the sperm is getting energy. So with this energy, what it is doing? It is making a move towards the oocyte. Yes, everybody. So this is what, it is the very first step that is called as your chemoattraction. Yes, everybody. Now your step 1 is done. It is the chemoattraction of the sperm to the egg by the soluble molecule. So what is the soluble molecule? It is going to be your, in case of your sea urchin, yes, it is going to be which one? Resac. Yes, everybody, come on. And that is going to bind with the bindin. So this is showing a species specific interaction and it is going to be timing specific. Yes, everybody. So this first point is done now. I think that now you are clear in that. Any doubts, please ping me. Right. Fine. So this is what, these are the steps that is taking place in the sea urchin. So now what happens? The sperm, it is going to contact this jelly layer. So where the jelly layer is present? In the egg. Okay. Then the second process takes place. So what is going to be the second process? It is the acrosomal reaction. So what do you mean by this acrosomal reaction? So there is going to be a calcium mediated fusion. Okay. So what is this acrosomal reaction? So there is going to be a calcium mediated fusion. Okay, so these acrosomal mediated species specific recognition is going to take place by means of this binding. right? So once this is being done, then it is going to lead to the next stage. Okay, so the first stage, it is the attraction, it is done. So what is going to be the second stage? So your acrosomal vesicle is going to undergo the exocytosis, right? So what do you mean by exocytosis? Yes, what do you mean by this endocytosis? Yes, endo means coming inside, exo means it is being sent outside, isn't it? So that is what it is called as exocytosis. So what it is going to do? It is going to release the enzymes. So the enzymes that are present within the acrosomal vesicles, it is going to get pushed out. Okay, so that is why it is called as the exocytosis. Yes, everybody, I hope that this is clear. Right, so your acrosomal vesicle, it is going to get burst and it is going to release all the enzymes. Remember, only at that time this binding, it is going to get exposed. Okay, so till that time there will be there will be the chemo attraction. Okay, by means of the resac. So your sperm and the egg, it will get attracted, right? But the original binding, okay, so this egg is going to attract the sperm, right? So that will take place. So the sperm will be near the egg, but this original binding, when it will take place after the exocytosis of the acrosomal vesicle. So that is why it is called as your acrosome reacted sperms. Okay, so after this acrosome is releasing the enzymes, the binding receptor, it is going to get exposed. Okay, so to that binding receptor only your this fellow your resac is going to come off and bind with, right? So this is a very important process that is taking place during your molecular fertilization, right? So next, what is happening? See, in the acrosomal reaction, it is going to be a calcium mediated fusion. Okay, so now only your species specific recognition by binding is being allowed, right? So this order is very important. First one, it may make a contact with the jelly layer. Then there should be this acrosomal reaction. After the acrosomal reaction only, there will be the binding between your resac and binding, right? Third, now the acrosomal enzymes are released. So what these enzymes will do? It will make a path. It is going to digest the jelly layer. Remember, this sea urchin, it is an invertebrate model. Okay, so that is why you are having your jelly. So if it is going to be this vertebrate, yes, then what you will have? Yes, everybody, come on. Vertebrate, it is going to have your zona pellucida, isn't it? Right. So that is the difference between these two. Okay, so these vertebrates, they are going to have this zona pellucida, right? Fine. So then what is going to happen? Your jelly layer is getting digested. Then there will be the binding exactly to this vitelline envelope. Okay, so it is going to make a bind with the vitelline envelope, right? So once that is bound, then there will be the final fusion of your acrosomal process membrane and the egg membrane. Once this fusion is taking place, all the things will take place, right? So this is the overall mechanism of the fertilization steps, right? So the in case of your acrosomal reaction, you have to remember the key word, that is going to be your calcium mediated fusion, right? And then this is going to release many enzymes like your metalloproteases. So they are going to digest your jelly layer, and then it is going to make a binding with the vitelline envelope. So once it is bound with this vitelline envelope, it is now made free to undergo the further fusion. So the membrane of the egg and the sperm, it is going to get done. Okay, so for this, the sperm has to reach the inside of the egg layer. Okay, so if you take, even though the sperm is having a very good motility, okay, so it is not enough. Why? Because the distance is very big. So even though the sperm is traveling that fast, even though it is having this ATP and this mitochondrial ATP is giving the energy, the sperm cannot reach the egg. Then how it will reach? See, the reproductive tract of the female mammals. Okay, so this is playing a very important role in the fertilization, right? So this is a very important point. Already your CSIR net question has come from this point, right? So this reproductive tract, especially for the female mammals, they play a role in the fertilization. Okay, yes, so that is what it is making a very good contribution, right? So even though the sperm is swimming very fast, okay, so they have tested in mice, hamsters, guinea pigs, cows and all, no, but no, these, even this fastest running sperm, they cannot move based upon their own power, but it is transported only by means of this oviduct. How? The muscular activity of uterus, it is seriously important to transport the sperm inside. Right, everybody? So, so for to make the sperm to be competent enough to undergo the fertilization, it must undergo a process called as the capacitation. Yes, everybody? So this capacitation means it is a set of physiological changes. So that is going to allow the sperm to become competent enough, right? So that is a very important definition. So this definition is very directly already came up, in your part B of CSIR, right? Okay, so you should be very careful. So all these concepts are very, very direct, and you will be getting conceptual based questions from this topic, right? So don't miss out any of these, right? So what do you mean by what do you mean by this capacitization, right? So it is a set of the physiological changes. So it is going to allow, right, the sperm to be competent enough, okay, to fertilize the egg. So that is what it is being called as the capacitization. So how then if at all there is no reproductive tract, right? Fine. So you imagine that you are head of this IVF center, okay? So you cannot produce any reproductive tract for your in vitro fertilization. So in that condition, what you will do? You are going to mimic the condition. So what condition? You will be providing with the calcium ions. You will be providing them with the bicarbonate. You will give the serum albumin. Okay. So all these things, it is going to compensate the role that is being played by the reproductive tract, right? So what are the important changes that are taking place during your capacitation, right? So this is very important. Number one, see the fluidity. Okay, so this fluidity of the sperm plasma membrane, okay, so it is going to get altered. Why altered? See, the cholesterol is being removed. Okay, so already in the other classes we could have seen how your membrane, it works with the cholesterol. Okay, so if at all the cholesterol is being removed by means of this albumin protein, yes, then it is going to affect the fluidity, right? Okay, so it is not, if at all you are adding the cholesterol, okay, so you must actually remove the cholesterol. By chance, if at all you are adding the cholesterol, then there won't be any capacitization. Yes, done. Okay, so the second point is, so what is the first point? First point I told you is about the cholesterol. So this cholesterol should be removed from the sperm. How you are removing the cholesterol? By adding this albumin, right? So the second one, yes everyone. So the second one, it is the certain carbohydrates, okay, or else the particular proteins, it is going to get lost. Okay, so why this protein must be lost? See, these compounds, it may block the recognition. Okay, so I told you that if at all it is going to be the invertebrate, you have your jelly, right? And then you have your vitelline, right? And in case of the higher vertebrates, it is going to be the zona pellucida. So the zona pellucida, it is going to contain a binding site. There may be certain carbohydrates or protein that is going to block the recognition. So you should remove all those proteins. So that is what taking place in the second step of the capacitization. Right? Okay. So the third step. So what is the third step of the capacitization? see, the membrane potential of the sperm, it becomes negative. Okay, so I'll tell you a lot on this membrane potential and how this is going to prevent this polyspermy. Right. So now, as of now, what happens? Already the sperm is matured enough and it is going to bind with the oocyte. So at this condition, this membrane potential is becoming negative. Okay. So the membrane potential of the sperm is becoming negative. Why? Because your potassium ions are leaving the sperm. So that is why it is becoming negative. Right. And then the calcium is going to enter the sperm. Right? Okay. So this calcium as well as this bicarbonate ions, yes, so they are very critical to activate your cyclic AMP. So this cyclic AMP, it is going to facilitate your membrane fusion. So that is what it is, the ultimatum of your acrosomal reaction. Right. So fourth one, it is going to be the phosphorylation. Right. So what are the major points? Shall we see? Right. So the first one that we have seen, it is going to be which one? Yes. So under the capacitization, I told you the first important step of the capacitization. Yes. What is it going to be that? Yes, yes. So if at all the sperm must mature, what should happen? Removal of cholesterol. Right. So how it is going to get removed? By means of this albumin. Right. And then second, so there is a removal of certain protein or carbohydrate that is going to block off your zona pellucida. Right. Third one, the charge, isn't it? Because your K plus ions is coming inside, it is going to create a negative charge. Right. And the fourth one is because of the phosphorylation. So these four steps are the key steps of the capacitization. So if at all you are performing your in vitro fertilization, then what you can do? So there is this is all taking place in the reproductive tract of the female. Okay. So if at all you are carrying over the in vitro fertilization, maybe you cannot produce a reproductive tract. Instead, you can use the albumin. You can add this calcium ions. You can add and this potassium, right? So that is what I was telling you here. So if at all you want to do it in vitro, you can add all these things. You can add the calcium ions, bicarbonate, right? And then your serum albumin, such that you will be able to achieve the same thing in this in vitro condition. Yes, everybody. So this is a very important process of this capacitization. Right. So now once this capacitization is doing, it is serving as a block for the polyspermy. Right. So that is the next important step. Right. So see this condition. If at all this isthmus is excised, so there is increased amount of the polyspermy. So what does it mean? It means that this isthmus is required for the polyspermy prevention. Right? Okay. And shall we see a question for our better understanding? Yes, everybody? Right. So come on. So this question is from June 2012. Right. So in an experiment, the sperm was removed from this epididymis of the male mouse. Right. And it was added in a dish. So that is going to contain appropriate media and oocyte. Okay. Now, see, sperm is there and then oocyte is there. Even though both are there is no fertilization. Right. But if you take the sperm and place it directly in the uterus, then that mice became pregnant. Right. So what does this suggest? So does it mean that the sperm, it have to travel some distance such that it is going to attain the fertilization ability, or the oocyte is secreting some biochemicals or factor that is going to help them to fertilize, or the hormones in the body, it is helping the sperm to fertilize, or the content of the reproductive tract. See, 4 marks for this question. The content of the reproductive tract, it is interacting with and activating the sperm. Yes, everybody. So which is the correct answer? And just now we saw all these important points. Yes. So which is going to be the correct answer? Yes. No doubt. Ultimately, it is going to be your option 4, isn't it? So, just now I told you that female reproductive tract, it is playing an important role in the capacitization. Yes, so that was the total story I was telling you about. Yes, you remember that? So, no, your sperm, it is going to get activated, no, one and only if this female reproductive tract is there. Okay, so this female reproductive tract, it is what it is playing a role, and that is what it is going to lead to the capacitization. Yes, everybody. So you got the trick behind this question. This is one of the important question, and definitely you should have a detailed look on these concepts. Right? Okay. So see one more question from December. Right. So the capacitization of the sperms in human, where do they occur? So they occur during this copulation, or they occur after the acrosomal reaction, or they take place in this ampulla of oviduct, or they take place in this epidermis of the testis. Yes. So they take place definitely in the ampulla of this oviduct, isn't it? Yes, so that is the place where the fertilization is also taking place. Right. Very good. So shall we make a move into the December 2013 question? Yes, everybody. Come on. So the next question says that the capacitization of this mammalian sperm, it allows them to be activated within the uterus, and it is going to facilitate the fertilization. Okay, so the following statements were made regarding the events that occur during the capacitization. Okay, so the removal of cholesterol, right? The removal of non-covalently bound glycoproteins, increased expression of fibronectin, decreased permeability of the calcium ions. So you have to identify with the correct answers. Okay, so which they are correct. So which option it is going to be the correct one? Yes. So cholesterol removal. Yes, it is there. How do you remove the cholesterol? By adding this albumin, isn't it? So definitely, you know that A is there, right? So the option without A you can eliminate, right? Then coming to the option B, removal of this non-covalently bound glycoprotein. Yes, of course it is also there, right? So definitely your option B is also there, right? So you can eliminate this option. Now coming to the next one. So increased expression of your fibronectin. Yes, and that is also going to be a true statement. But this decreased permeability of calcium, why decreased? There should be the increased permeability of calcium, isn't it? So obviously this is going to be the correct answer. Yes, everybody, I hope that now you got to know about this capacitization and how an important topic it is. Yes, okay, fine. See one more question. This is from your December 14. Now I think almost for 3 consecutive years, to 12, 13, 14, it is very repeatedly occurring. Even in 18 we have. I'm just telling you, always every year by year and question paper by question paper, definitely one question is there regarding this fertilization. Right? Okay, so we will have a look at this question. For the successful fertilization in the sea urchin, the interaction between the surface of the egg and the acrosomal protein, specifically, there is a 30.5 kilodalton protein called as bindin is necessary. Okay, right. So the following factors, they could affect this interaction, and it is going to prevent the fertilization. So there is a removal of your gel polysaccharide, or yes. So what will is going to affect the interaction and which is going to prevent the fertilization. Read the question properly. So this is going to prevent the fertilization. So if you remove the egg jelly, what is going to happen? Or if at all you are going to remove the bind-in receptor. Okay, so what is going to happen? So bind-in receptor, where it is present? So it is present in the egg jelly, or it is present on your egg vitelline membrane, right? And the final one, it is that removal of the bind-in receptor from the single cluster on the vitelline membrane. So which is going to be the correct option for this particular thing, right? So which is going to prevent the fertilization, right? So if you see that, yeah, so which of the following is really going to affect this fertilization? Exactly. So now we have already seen that your bind-in, your resac is being given by your oocyte, right? And bind-in. So this bind-in is given by whom? It is given by the sperm. Okay, right. So now that part you will be clear, right? So if you remove, okay, if you remove them, what is going to happen? So if at all you are going to remove the bind-in receptor, okay, on this egg membrane, then it is going to prevent the fertilization. So what is this bind-in receptor? That is nothing but your resac. Yes, so bind-in is present in the sperm and this bind-in receptor is present in the egg, right? Likewise, if you take this egg jelly, okay, so if you remove your egg jelly polysaccharide, even that is going to prevent the fertilization, right everybody? So this correct answer is going to be answer C with your A and B option, right? Okay, so see one more question from 2015. I told you, like every year, so even today we have seen this many questions. So we started with 12, okay, so June 12, December 12, and then December 13, December 14, and December 15 also you are getting a question from the very same topic. You can understand the importance of this topic by means of this question. Yes, yes Sachin, come on. Absolutely right. So the next question says that your successful fertilization, yes, in the sea urchin, it is going to demand the specific interaction between the protein and the receptors. Okay, so which is going to be correct? Right. Okay, so which is going to be correct? Right. So what the egg is going to contain? Yeah, egg is going to contain this resac, right? And the sperm is going to contain this bind-in, right? Okay, so this bind-in, this resac, it is called as the bind-in receptor. Why? Because it is going to bind with the bind-in. This bind-in is going to act as a ligand. Okay, so this is behaving like a bind-in receptor. Yes, now you got it? Fine, absolutely. Yes, Sachin. So like now what is going to happen? Yeah, so which of the following combination is correct? Yes, so now we know that your this thing, your second option is going to be wrong definitely. Okay, and not probably the fourth option also. So you will have a doubt between 1 and 3. Right, so here it is saying that your bind-in is present in the acrosomes and the bind-in receptors are present in the egg vitelline membrane. Right, so this is going to be the correct answer. Here it says that the resac is on your egg jelly and then this bind-in is on the sperm membrane. I told you that this bind-in is going to be present deep inside. Okay, so only after this acrosomal reaction only it is going to get exposed, right? So you got it? Yes. So the correct answer is going to be your answer 1. Yes everybody? Yes Sachin, I hope that now it is clear to you. Yes. So no, this bind-in, it is present deep inside. So only after this acrosomal reaction has taken place and all the enzymes has been released, then only this bind-in will be exposed. Right? So now it is clear to you. The correct answer is going to be your answer 1. Yes everybody? Yes Sachin, that's great. Come on. So let us have a look between this monospermy as well as this polyspermy. Monospermy means only one sperm and the egg is going to fertilize. But if at all the entry of the next sperm is not prohibited, it is going to lead to this polyspermy. See, this polyspermy, it is highly disastrous condition. Why it is disastrous? Because it is going to lead to the very, very tedious condition. So it may lead to the formation of merocyte. What is this merocyte? See, many sperms may be there, so they will die off in the cytoplasm of the egg. Okay, so you have your one sperm and the egg, it will be fusing, but all the remaining sperms that are entering, it will be die up in the cytoplasm, leading to the merocyte. Right? Okay. Or else it may lead to this pathological polyspermy. So what is that? Here the nucleus of the sperm is getting to fuse with the nucleus of the egg. Right? So this is what ultimately it is taking place during the polyspermy. Right? So there are different methods to prevent the polyspermy. So definitely we will be seeing that. Yes. So this is a little bit information on how our courses are going on at our Unacademy Plus. Right? So our Unacademy Plus, it is a very good platform for you to learn a lot on your lessons. Right? So currently I am conducting 2 of the classes. One I am having a complete course on the life science. That course is commencing at 10 a.m. every day, and one more course I am conducting is the practice session. So full MCQs, live MCQs, you can participate in this online quizzes, right? So on live you will get instantly, you can have access, and you will have a training on these attempting these sessions. So this is what last month we were dealing with each and every subtopic of your unit 13, that is the methods. Right? And this month I have just started with your cellular biology. So I am done with A, B, so tomorrow I will be dealing with C. So this is what I am conducting at our Unacademy Plus. So seriously, I'll tell you, this platform is highly engaged, and I see a lot of students are participating every day in these courses, and many of them are getting into these paid courses, and it is making a very good transformation in the way they are studying. So apart from that, apart from these live classes, if at all you are preparing for your upcoming GATE examination, so you want to study, very quickly this fundamental process, that is nothing but your molecular biology, or you want to study this developmental biology for any other exams, or the methods. So all these recorded versions are also available. So these are conducted for your 19th, December 19th batch. Okay, so all these informations are already available for you. Right. So if you think of the fees, yes, the fees is actually very much reduced. Yes, and it is going to be very less for your program. Yes. So the 6 months package, it is getting charged only 8.4, and you can definitely use my code Saranya Live. With this you will be getting a 10% off on the total fees. Right? Yes. And then if you subscribe for the 12-month package, it is very cost effective, only 10,500, and post discount it is only 9,450. So don't forget to use the code Saranya Live such that you will get these discounts. Yes, so our platform is seriously very helpful to you, right? And you can think of first attending up the live classes. So these are all called as the special classes. So once you come up at our Unacademy platform, right? So you can enroll yourself with these live classes. Okay, so these are all for free, and tomorrow I am taking up an important techniques for life science. Okay, so you can enroll yourself for that course for free, and you let you experience and let me know how was the experience. So this is a testimony that has been given by many of my students. So you can see that most of them they have qualified this examination. Yes, so it is seriously it is transforming the way of how they are learning, right? So don't miss out this chance. So let you be in this list of successful qualification, yes, such that you can make a very big transformation in the way that you are studying. Yes, everybody, so cheer up. So let us continue on our understanding on the prevention of polyspermy. Right, yes everyone, that's great. So you can you can have a look at it. Yes, so know that if you take of this polyspermy prevention, it is of 2 types. One, it is going to create a slow block, right? The next one, it is going to be the fast block. So these 2 are the very important types, so that is taking place. Yes, everybody? Right. Oh, yes. I'm sorry. Right. So the first block, it is called as the fast block. So what do you mean by this fast block? right? So this fast block, it is immediate block. Okay, so how immediately this blocking is taking place? See that it is going to change the electric potential of the plasma membrane. Okay, so I'll tell you, we have already heard a thing saying that only the opposite poles attract. Yes, have you heard it? Opposite poles, it will going to attract each other. You have heard this, isn't it? So the same thing applies for your egg and the sperm. Usually if you see, right, so usually the charge of your egg, okay, so it is minus 70 microvolt, right? Fine. Now what happens? The sperm, it is going to contain a positive charge. So that is why both these are going to attract and influence each other, and it is going to bind with each other. Now once a single sperm has bound, yes, immediately there is going to be a change in this resting potential, right? So now what is going to happen? See, this minus 70 microvolt, it is getting converted into plus. So what is the charge of the sperm? Already I told you that the sperm is positively charged. Now this egg, it is becoming positively charged. So the same charge will attract each other? No. So it is going to repel each other. So this is what it is called as your fast block. Yes, everybody? So you got the point now? Right. So we will summarize it very quickly. What is going to be the fast block? Right. So the first one, I told you that the fast block is because of the charge. Right. So if you take the egg, it is going to be minus 70 microvolt. And the sperm, it is going to be positive. So once the first sperm binds, now what is going to take place? There is going to be an alteration in the charge. Now the egg is going to become plus 20 microvolt, right? And the sperm is already positive. So these two, they are going to repel each other, and that is what it is called as the fast block. Right? Okay, so this is what it is important. So one of the scientists, Jaffne, a very interesting scientist. So what he did, see that he want to test whether this resting potential is there, or what sort of thing is taking place with this. So what he did, he found that if at all after a sperm and a egg has bound, if at all he is supplying the electric current to make them negative. Okay, so I told you that the sperm is already positive. Your egg was negative. Now after the first sperm binding, it is converted to be positive. Now if you are artificially giving the negative charge to it, okay, so now the egg becomes negative, right? So the sperm is positive, right? So now what will happen? Again the fusion will take place. So that was his experiment. Whenever you are artificially supplying them with the negative charge, right? So there was a polyspermy. Why? You have made them to be the opposite charge. If there is an opposite charge, it is going to attract each other. Yes, everybody? So this is what it is taking place in the fast block. For that, there is a voltage sensitive component. So that is a positively charged fusogenic protein. Okay, so that is what it is regulating the charges. Right? Okay. So you can see that if at all you are increasing or you are decreasing with your Na plus, okay, then you are increasing the percentage of the of the polyspermy, right? If your Na is very less, right, then there is a increased chance for the polyspermy. Very important point, right? So your sodium ions, they are, if at all their concentration is too much decreased, so that is leading to the increased percentage of the polyspermy. Yes, fine. So now the next important point is, after the fast block, it is going to be the slow block. So how the slow block is taking place? See, the fast block is very transient, okay, so that is a temporary one. But the slow block, it is going to be a permanent one. Like if at all you are having a tap repair at your home, so temporarily you will put some cloth and lock it, right? But then permanently a plumber will come and fix it. Likewise, what this is being done during the polyspermy prevention, for the swift reply you are making the conversion of the charges. Okay, but this is going to be very for a short time. Okay, then it will come back to this original charge. Within that, this cortical reaction should take place. Okay, so that is what it is done by the slow block. Okay, so what is going to be the slow block? It is called as the cortical granule reaction. Okay, so this cortical granule reaction is what it is called as the slow block. So now what is going to take place? See, so what are these cortical granules? So where is the acrosome present? Don't get confused. This acrosome are present in the sperm. Right? Fine. So they are also rich in the enzymes. Right. But these these cortical granules, they are present in this egg. Okay. So they are also rich in the enzymes. So where they are located, see, almost 15,000 cortical granules are present beneath your egg plasma membrane, right? So that is what. So whenever a sperm has entered, these cortical granules, it will fuse with the plasma membrane and it is going to release everything, right? So that is what it is taking place by your cortical granule exocytosis. So there it is going to be your acrosomal reaction. So even that is going to be an exocytosis in the sperm. Likewise, after this acrosomal reaction, the sperm is binding with the oocyte. Now this is going to induce a cortical granular exocytosis. Okay, so it is going to release so much of enzyme. What are they? Number one, these proteases. So this is what, no, it is the product of the cortical granule. So it is going to be the proteases. So the proteases, it is going to dissolve, your vitelline envelope. Okay, so they will be clipping off all the binding receptor. You know that the binding receptor, it is the place where this binding is going to come and bind. Okay, so if already one sperm has bound, right, so then it is going to release the proteases and then this binding receptor is now removed. So now if at all many sperms are coming also, it cannot bind. Why it cannot bind? This receptor is now being clipped off. Yes, everybody? So this is the thing that is taking place in the slow block, right? So apart from the proteases, they will be releasing these mucopolysaccharides, right? So they will be leading to the next stage. So they will form the fertilization envelope and then they will make it ready for the next stages. Probably it will prepare the cell for the cleavage. Okay, so all these things are taking place in your slow block. Yes, everybody? So very quickly we will see. Yes. So we saw about the fertilization. So under which we saw the sea urchin fertilization. We saw about the binding of your resac and then this binding, right? So remember, where is the resac present? It is present in the egg jelly, right? Okay, so this binding fellow, he is present deep inside your acrosome, right? And then this resac is called as this binding receptor. Fine. And then what else we saw today? We saw about this capacitization process. So in this capacitization process, what is literally taking place? In this capacitization process, the cholesterol is getting removed, right? And all the different clipping protein and your glycoprotein, it is getting removed, right? So apart from that, what is taking place in the capacitization? Calcium influx is there, right? And then your potassium ions, so they are going to move out, right? So all these changes are taking place in the capacitization. So that will take place only in the female reproductive tract, right? Fine. And then we saw about how this actual fertilization is taking place. So the moment this your resac and binding or binding, so when this binding will be exposed only after this acrosomal reaction. Okay, and once this is being done, then we had a look at the polyspermy prevention. So under the polyspermy, we saw about the fast block as well as the slow block. So fast block, it is the change in the charge. Slow block is your cortical granular reaction. Yes, everybody? So I think that this is all clear to you. So shall we take up a question? Yes, so before we are ending, we will take up a question for them. Right, so this is from June 2013. I told you, no, definitely one 4 mark and one 2 mark is there in the fertilization. Right, so the polyspermy, it results when 2 or more sperms they are going to fertilize an egg. Right, so it is usually lethal because it will result, in the blastomeres with, different numbers as well as the different types of your chromosomes. Okay, so many species they have 2 blocks, of course, and we have seen the best example with the sea urchin. Right, so now you are going to tell me the correct answer. Come on everyone. Yes, so a first block is immediate and it is going to cause the egg membrane resting potential to rise, so which does not allow the sperm to fuse with the egg, and that is being mediated by the influx of the sodium ions. Yes, okay. So the first block is immediate and it is going to cause the egg membrane resting potential to rise. So it is not going to allow, the sperm to fuse with the egg, and it is going to be mediated by means of the efflux of the sodium ion. Just think of, it is going to be influx or efflux. Definitely it is an influx of the sodium ion. Okay, so by that only we will eliminate this option. Okay, so the slow block or the cortical granular reaction is mediated by definitely the calcium ions, isn't it? So definitely you know that A and C, your option 1 is going to be right. C, 4 marks for this question. Yes everybody, so you got know how to solve such question, very direct, and all these it is very directly coming off from your examination. Yes everyone, fine. So that's really great of you all. Thank you everybody for your patient listening. Right, so please do think on our courses at your Unacademy. So definitely you will get a lot, you will learn a lot of information from us. So don't miss out. So already many students have cracked, so why not you, isn't it? So definitely it is possible to crack with this. Yes, so you can give a try by enrolling yourselves at the special classes. So they are free, and tomorrow I do have a class at 8 p.m. in our platform. So you can very well come to our platform and enroll yourself, and you can think of enrolling with our 1-year program or 6-month program. So if you use this code, you will get a 10% off. Yes everybody, I hope that this is all clear to you. Yes, so definitely I will catch you up in another interesting session. All the very best. Bye bye and good night.